states this is the applicant has to be from a European country.
Also applies to Taiwan.
Has to go to a European country also.
But Taiwan has become a European country.
Yeah. Maybe not the best time, but I mean, actually, when I do this
Good morning, ladies and gentlemen. We are about to begin our conference today. So please find yourself a comfortable seat. Jordan, Professor, and distinguished speakers and our guests. I'm very honored to welcome everybody to come to the 2013 conference on the ELSI of biopharmaceutical research. On behalf of the ELSI group of NRPB and the National Taiwan University Biotech Center, and also the Department of Social Medicine, College of Medicine, NTU. We are very honored to organize and prepare this small conference. And we have a distinguished professor from France and also our local, our domestic ELSI scholars. We plan to join and to share our research ideas in ethics law and social impact on research ethics issues. And in this conference, Professor Jordan will give his keynote, and then later our participant research will present their research papers from different perspectives. In the past 2 years, the ELSI group of NRPB has been devoted to improving better ethic and legal regulatory framework for biopharma research. And at the same time, equally or even more important is promoting the public consultation, deliberation, and also communication and education. So for that purpose, the public engagement group has been devoted to communicating with the general public and also the focus patient groups through different type of workshops or focus group meeting. And we know that in past few years, there has been quite a lot of advancement in our medical research, as well as the IRB regulation and legal framework and legal legislation increased a lot for better self-protecting human subject research in Taiwan. And there have been lots of interesting debates and also some dilemmas generated in this context. So today we have different topics focusing on research ethics and law, and also some research paper concerning the public consultation process and the general public's their opinion toward medical research. So we expect to have a very fruitful exchange through today's conference. So on behalf of the organizing bodies, I express our utmost welcome to you all. And so we are moving on to our first speakers. And I'm very honored to introduce Professor Xie Fengzhou, formerly Professor Dai Hua. He will be chairing this the next section. His good friend, Professor Jordan. But Professor Dai Hua, because he has had some urgent issues, urgent meeting in Chengda, so he won't be able to be here. And at the last minute, my mentor, Professor Xie Fengzhou, actually saved me, so he is willing to chair these sessions for us. Let me do a little introduction for Professor Xie Fengzhou. Xie Fengzhou, he is actually the co-founder, founder of our NTU Biotech Center. And he also founded many important research centers in NTU. He has been very famous for his innovative ideas and stimulating comments, encouraging our young generation to do something different. So every time I met him, I was quite often kind of 被他骂. kind of accused or criticized by his critical points. But we know that his encouraging and challenging opinions quite often help us move forward to the next era. So let's welcome Professor Chen to chair this session.
I'm the 这个叫做救援投手。Okay, but today I'm very happy that just last week Dr. Cai Buchang get promoted to full professor. Okay, so let's give him a big hand. And he told me he'll prepare a big banquet for us for celebration. Okay, I think for him it's a very important landmark. In the medical campus, usually what is important is the cure of physical disease. Okay, so for because Buchang is deal with something very abstract like ethics or integrity or law, or actually it's not important. Actually I know about the words of integrity until I am 60 years old. Before that, I don't know these words. Okay, so I think his promotion becomes I think there will be very significant impact because he is now has a good position to speak up for the right things. So I encourage him, and maybe also our coming university president, Professor Yang. I think I told him that the next stage, the important things in Taiwan might not be nature or science paper, but the social justice. So he think I'm crazy, but I think that's what I've heard. So I think I just surprised to see the topics of today is involve the prenatal diagnosis. I think it's a messy issue now. Okay, actually it is the major field of my past maybe 20 years, but we start nothing. Start from scratch the so-called maternal serum screening in Taiwan in about 1990. Okay, and 2 years has passed. Recently, I saw the statistics that is, every year we have about 180 Down syndrome baby were detected prenatally, and of course, most of them were terminated. And the living Down syndrome every year now maybe less than 10 in Taiwan. Okay, so what we do is first we promote amniocentesis. Okay, and then followed by 2-marker Down syndrome screening, and of course we become a 4-marker Down syndrome screening. Okay, I think that's enough to achieve this 90% detection. But now, of course, there is a new, commercial product like free DNA, okay, or CDH, microarray, something, but it becomes a very, commercial campaign. Okay, so it becomes a very, of course, some staff previous in our hospital. That's actually my students. I handpicked him to help to establish good DNA diagnostic facility for all the Taiwan. But unfortunately, it turned out to be a commercial scandal. And of course, I formally, accused him to the hospital authority. But unfortunately, there is no investigation at all. Okay, so I think this medical campus actually is, very shamed. because I think he has good connections through political connections and economic connections with different level of leaders in this campus. So even if I officially with a written document to accuse him of his misconduct, there is no investigation at all. Okay, so I think I'm very disappointed about. So I think I always encourage Fu Chang that ethics is not just oral service. You have to reinforce it. Okay, so that's why now that he is a professor, now he can speak without any too much consideration. But I think the student learns, they saw instead of they saw what really happens. Okay, instead of they learn, they listen what you talk. Okay, so I think because this prenatal diagnosis involves a very huge commercial interest. Okay, so I think I'm very glad that Professor Jordan will be talking about this, and I hope that maybe we can further elaborate this issue to even to the public setting, okay, through some different academic occasions or academic society to let the society has some correct understanding of this what will be happening with those so-called advancing. So I think perfect child is not possible. Even you can do all kinds of gene testing, because what serious I think what concern the parents most is something like a CP, right, and something like autism, something like learning difficulty, dyslexia, and hyperactivity. I think that cannot be tested through those DNA diagnoses. So what can as far as I know, even with the amniocentesis plus RACDH, the detection rate of abnormality maybe increase just a little bit. Okay, so for me, I think but actually it really changed things, because before we use DNA. Chromosome examination, we promote ultrasound screening for the so-called 20 weeks gestation. Actually, only with that we have eliminated very big spectrum of all congenital defects. Things like the congenital disease accounts for about 1% of congenital defects, but the difficult thing is severe things like single chamber. I think it can be easily found by ultrasound before birth, so it was eliminated. Okay, so we had just only ASD, VSD for the pediatricians to work up. Okay, so it actually changed the whole picture of the pediatric disease spectrum. So I think so everyone was very curious about what the DNA can do in the future. It's ready for the future parents, and of course there are lots of commercial promotions. Okay, so there's all kinds of connections between physicians and merchants. So I think today, Professor Jordan, the topics is just in time to give us some very, I think will be very productive insight into this issue. Thank you.
Thank you very much. Well, let's get this on. Okay, very good. So I'm not an ethicist. I'm a molecular biologist or a genomics person. So I'm not going to give conclusions, well, not many conclusions on what should be done. What I thought I wanted to do is to alert you to what is going on, because there's really a very fast technical progress due in particular to new generation sequencing, to the fact that it has become very easy and very inexpensive to sequence human DNA. And this is getting into clinical practice, and this is having impact in many fields. Of course, it's having a lot of positive impacts, but it's also raising a number of problems. And so I thought it would be interesting with this audience to tell you about the latest developments in the general field of prenatal diagnosis. So let's see if I can operate this. Yes. So new generation sequencing, you know, this is a kind of technical revolution that began about 5 or 6 years ago after the genome program was finished. The genome program was done with sort of conventional technology which had been invented around 1977. But starting in 2005, 2006, new systems began to appear, and okay, they are based on a lot of progress in electronics, optics, micromachining, and so on. And they essentially sequence millions of DNA molecules simultaneously. So they provide enormous amounts of data at relatively little expense. Initially, those were big machines. I mean, machines which typically cost $500,000 and which are able, in one session, to sequence to a good quality level a whole human genome, something which had previously taken 10 years, billions of US dollars, and thousands of scientists. So this is a site which gives you a world map of where these machines are located. I know this doesn't work, of course. Well, you see a lot in the US, of course. You see a big cluster in China, that's in Shenzhen, that's BGI. If we zoom in on Taiwan, actually there's quite a few of those systems in Taiwan, mostly in Taipei and around Taipei, of course. Okay, this is quite striking. This is the evolution of the cost to sequence one human genome over the years. Okay, the first sequence of the human genome, you can say it cost maybe $1 billion US. But by 2001, if you wanted to sequence another human genome, the cost was about $100 million US. The solid line is the decrease of the cost of computing. This is so-called Moore's law, where the cost of computing goes down by a factor of 2 every 18 months, which is very, very fast. I mean, it doesn't look so fast on this image just because of the scale, but just think about it. If Moore's law applied to cars, our cars would cost 50 cents and would run at the speed of light. So this Moore's law shows a very quick decline, and as you see, the decline of the cost to sequence one human genome more or less follows Moore's law until 2006, 2007. That's where the new machines began to be fairly widespread, and you see then it drops down quite dramatically. So, of course, it depends how you estimate how good the sequence must be and so on, but this is the cost for a reasonably good human genome sequence with estimated less than one error every 100,000 bases. And this is now down to maybe $5,000 or $6,000, and it's probably continuing to go down. There is yet another generation of machines around the corner, and it will probably reach the goal of $1,000 genome quite soon. Of course, that's just getting the sequence, then you have to analyze it, and that turns out, depending on how deeply you want to analyze it, that ends up costing more than $1,000. But anyway, so this means really in just maybe 10 years, the cost of sequencing has dropped incredibly. I mean, 1,000-fold. I mean, it's really quite incredible progress. So as you saw from the number of machines, there are centers doing this all over the world, either in big centers or in research labs, or now also in clinical labs. And so it is indeed beginning to have a strong impact in the clinic. It's used quite widely now for cancer therapy to find out which treatment is likely to be effective in a given patient by actually sequencing all or part of the DNA of a biopsy, of a tumor biopsy, to find out what's the problem in a child which has some problem which you cannot find a cause for. To follow emerging diseases, you may have noticed that now every time a new disease appears, we get the sequence of the agent almost overnight. But and this is my topic for today, so it also allows new approaches in the field of prenatal diagnosis and what you can call embryo choice. So that's what I want to show you. Okay, this is just one of many many papers, well, essentially that say that this is what I wanted to underline. Getting a whole genome sequence in medical care will be as commonplace as getting an annual physical exam, except that the sequence, you only need to have it once, and then you can mine it repeatedly as knowledge improves. So, first thing is prenatal diagnosis. So what is going on in prenatal diagnosis? The important thing which is going on is so-called non-invasive diagnostics. Now, of course, as you know, prenatal diagnosis is normally done by amniocentesis or chorionic villus sampling. These are invasive procedures, they are unpleasant, and they have a risk of provoking miscarriage, which is around 1%. Now, it turns out that if you take maternal, if you just take a blood sample from the mother during pregnancy, the plasma contains some DNA from the fetus. Not fetal cells, you're not looking for fetal cells, you're looking just for free DNA in the plasma. So in the plasma there is a small amount of DNA from the fetus. Of course there's also DNA from the mother, because you always have white blood cells that have lyse and have released some DNA. So it's a mixture. It's a mixture where the fetal DNA is usually around 10% of the total. But actually, if you just sequence without separation, without trying to separate, if you just sequence this DNA and analyze it by bioinformatics, you can actually isolate the contribution from the fetus. So you can actually already now do some kinds of prenatal diagnosis just on a blood sample from the mother, which of course is a lot more comfortable and a lot safer. And so this is done for trisomy testing for chromosome 21 and a couple of other chromosomes. And it's actually already commercially available, at least in the US, and quite successful. So the company which first introduced this kind of test is called Sequenom. There are 4 other companies in the US, and there are companies elsewhere. Actually, I believe BGI is also proposing this type of test. And so at this time, this is mostly done as a screening test for high-risk pregnancies. High risk meaning the mother is over 35 or has some history of problems. So it's quite a large population. And if the test by this method indicates trisomy, then you go for the invasive tests. But that means you avoid a lot of invasive tests. I think I have a price list. No. So this is actually probably the medical test that has been the most successful, the most quickly, because it has, of course, the great advantage that you avoid invasive procedure, and actually, so people who would not have done an invasive prenatal test are quite ready to do this test. Okay, there's some technical issues, but the latest results are really very good. Almost 100% sensitivity and almost 100% specificity. I think the latest that I saw by one of these companies on like 300 women, there was no false positive and no false negative in this whole sample. So it's getting really good. So it's already covered by insurance in the US, which is a big issue in that country, as you know. It may actually turn out to be extended to a general screening, which means 4 million births in the US, 750,000 high-risk pregnancies, but 4 million pregnancies. Okay, of course, there's a lot of legal action between the companies because each one is accusing the other of infringing on its patents. So the professional body, like the American College of Medical Genetics, is a little cautious on the quality control and whether this should be used as a preliminary screening or actually a test. But I mean, this is, I guess, a technical issue that will progress quite quickly as the methods improve. So this is a recent item in Nature Medicine, which shows the 4 companies doing this in the US. So they all look at actually 3 trisomies. They use slightly different methods. At the time of this paper, they had these figures for sensitivity and specificity, but those have got better now. Also, one of the strong points of this test is that it can be done quite early. And you know, in the US, it's getting more and more difficult to obtain pregnancy termination if you want. And so being able to do the test as early as possible is an asset. And you see the cost goes from less than $1,000 to almost $3,000. I need to get some water. I just need to open it. I got it. Okay, so yeah, that's actually a summary of the previous slide. So as I said, this is a very, very successful test, and I guess on the whole it's a progress. There's not, I don't think it really raises very difficult ethical issues, as long as it's properly done, of course, and as long as it's available to the people who need it. Now this can be extended. I mean, if you are actually sequencing this mixture of maternal and fetal DNA, if you're willing to sequence deeply enough, I mean, to have sufficient accuracy and can do more analysis, you can actually go a lot beyond just looking at trisomies, at whole chromosome trisomies. You can look at partial duplications, and you can imagine that it will be possible to actually look at single mutations. So that you should be able, with enough analysis, to actually do prenatal diagnosis for any kind of mutation in the DNA. So I would predict that maybe in 2 or 3 years' time, we will see the announcement of the possibility of doing prenatal diagnosis for, let's say, cystic fibrosis using this method, non-invasive prenatal diagnosis for cystic fibrosis, which again is progress. So we can expect that because this is the ELSI issues that came out of a discussion from a biology bioethics panel at a recent genomics meeting in the US. So of course one can expect that there will be a lot more prenatal testing now that it's safe and not inconvenient and reasonably inexpensive. Then what is going to happen, as in other clinical fields where sequencing is having an impact, is that okay, you find what you are looking for. For example, you find if there is or there isn't a mutation in the cystic fibrosis gene, but actually you get a lot more data, and in this data you can have so-called unanticipated findings. You find variants of unknown significance. You find a deletion somewhere in the genome, and then you wonder, does this have a health impact or doesn't it? And in some cases you have data to say that this maybe have consequences. In some other cases you don't have data. Then you have all the issues. What do you do about it? Do you tell the patient? Do you tell the doctor? So that means when people go into this kind of testing, they have to provide some kind of informed consent, and the informed consent has to include the possibility that there will be unanticipated findings, and so people have to decide in advance if they want to know about these anticipated findings, which means you have to explain to them before they sign the form. So it actually raises a lot of issues. And then there are also the issues of parents' rights versus the child's rights. There are cases where you can have conflict between the two. And then of course you can also have social pressures. If by this kind of testing you find a certain kind of anomaly in the child, which means that if the child is born, he will be in a way a burden on society. If the parents want to keep the child, well, whose decision it is, does the interests of society come in which way, and so on and so forth. These are not really new problems, but they're just going to be more, to come up a lot more often, because there will be probably a lot more prenatal testing in the future, and because we will get a lot more information for each test. Okay, so this is non-invasive prenatal testing. Now I move to a slightly different issue, which is actually a lot more problematic, and this I can call prenatal checking for identity. What it means is non-invasive paternity testing, non-invasive prenatal paternity testing. So, in other words, your partner is pregnant and you want to know if you are the father. Or she wants to know who is the father. And you just send a sample to this company or any number of other companies who do this type of testing. And actually all you need is a sample of the mother's blood and some kind of sample from the putative father, like a hairbrush or a toothbrush. You don't even need to have a blood sample, so he doesn't necessarily have to know that you are doing the test. And actually, this company, I think the cost is like $1,000, and that includes testing 3 potential fathers. So how does this work? This is actually this is technically a very easy test, because what you do again is you have the mother's blood sample, and in this sample you have some little fetal DNA mixed with mother's DNA. What you do is you use a microarray. This doesn't even need sequencing. You use a microarray that looks at maybe 100,000 SNPs, 100,000 variable points in the genome. At the same time, from the same blood sample, you can isolate mother's leukocytes, so you can find out which alleles of the SNPs are present in the mother's DNA. And so the ones which you find in the fetal DNA, in the mixed sample, which are not from the mother, they must be from the father. And then you just compare these with the DNA samples you have from the possible fathers. So it's actually very easy and very straightforward technically. But it's, of course, far from straightforward from the point of view of ethics and society. So you can imagine pressure from the official father who has some doubts and wants to make sure that the child is, the fetus is from him. And if the test shows that he is not the father, he may then pressure the mother to go into pregnancy termination. So I mean, it's a pretty lousy situation. But this is perfectly legal in the US. It is illegal, for example, in my country, because in France you are not allowed to do a DNA test to take the initiative. It has to be prescribed by a judge or by a doctor, and there have to be serious medical reasons for this. But of course, you could still send your sample to a company in the US and get the results. It would have no legal value, but you would still get the information. But in principle, in France, you can be punished quite strongly. I think it's 3 years in prison and 10,000 euro fine. But as far as I know, this has never happened. Okay, so that's, as I said, this is a technically easy but ethically very problematic application. And now the last part of my talk is how do you make sure you're going to get a high quality child? So not just to avoid this or that disease, but how do you ensure the best quality? And well, there are 2 ways you can do it. One is already available. The other one, well, I think will become available in the near future. So one way is just to be prudent and to test what mutations you and your partner may be carrying. So it's now quite possible to check for hundreds of recessive mutations for single gene diseases, Mendelian diseases, and so to find out if you and your partner maybe are both carriers for some rare, serious disease. So, in essence, you check your quality and your partner's genetic quality before embarking on having a child. And if you find out that you are both carriers for a given disease, then you can, if you still want to have a child other than by adoption, then you can go for in vitro fertilization and pre-implantation diagnosis for that particular disease. And I mean, and there is experience in doing this within the community, which is Tay-Sachs disease in the Ashkenazi Jewish population, where for historical reasons the corresponding mutation had a high frequency, and the disease, which is a pretty horrible neurodegenerative disease which leads to the death of the child at 2 years of age. So this was actually screened within the community with quite an interesting system. Maybe if you want, we can discuss it later. And this has essentially eliminated this disease from that community. Of course, there are this borders on eugenics, and this raises S issues. But if it's properly handled, it can probably be positive. But in this case, we're just talking about individual decisions, not collective ones. So again, there are companies that already provide this service. You see, carrier screening, Good Start Genetics. So they provide screening for more than 100 mutations. It has probably increased by now, and I think this is another. There are a number of those now where you can get yourself checked for a number of possible problems. Okay, so that's one way. But again, this is avoiding diseases, and this is a little complicated, but there will be probably a way to actually select the best possible child. And this is using something which is already in practice, pre-implantation diagnostics. So it is done at this time. So again, just to remind you what we're talking about, you do in vitro fertilization. You get something like 10 embryos usually, and you can actually let these embryos grow to a blastomerous stage where they have, I think, 8 or 16 cells. At that stage, the cells are still undifferentiated, so you can remove 1 or 2 cells, and you can extract the very small amount of DNA that you have in these cells and test the DNA. And so this is currently done in the case where both parents are carrier for a severe disease, like again cystic fibrosis, and so it's technically possible, not terribly easy, but it has been technically possible for quite a few years already to test this minute amount of DNA to find if the embryo is homozygous for the disease or heterozygous or completely normal, which is what's going to happen if you have, let's say, cystic fibrosis. You will have on average one embryo who has both normal alleles, 2 embryos which have one normal, which are heterozygous, and 1 embryo who is homozygous. So then in that case, then you do this testing on a number of embryos, and you take the 1 or 2 which are where the mutation is not present, and you implant those. And so you allow this couple, which would otherwise be at risk for a very serious disease, you allow them to have a normal child. So this is done. It's done in some countries. For example, it's not allowed in Germany. It is allowed in France, providing that the disease is serious. Of course, the definition of serious is something else. And just what I wanted to underline is that it is now conceivable to extend this rather than just looking at 1 or maybe 2 genes in the embryo to really get a look at the whole genetic constitution of the embryo. Because there are a number of groups working on getting a complete DNA sequence just from a single cell. And this is actually beginning to work. There's actually 2 ways of doing it. One way is to amplify the DNA and then sequence it. And there the difficulty is that you are starting with a very, very small amount of DNA, so you're amplifying very, very widely, a millionfold more than a millionfold. And so you need the amplification method to be very uniform. If it tends to amplify some regions more strongly than others, over many, many cycles, it's going to give you an enormous difference. And so then when you sequence, you're going to sequence some parts of the genome 1 million times and other parts only 1 or 2 times. And so you're not going to get data covering the whole genome. So a lot of the effort, the technical effort, is at finding new kinds of amplification methods that give you very even amplification over the whole genome. And this has made a lot of progress. This is a citation from this paper. I don't remember what the acronym MALBAC stands for, but it's some sophisticated way of amplifying the genome that gives much better uniformity than PCR. And this is a quote from the paper, and they say that they got, well, pretty good genome coverage, and they were able to identify individual single nucleotide variations, which means they can identify mutations in the whole DNA. So this is one way of doing it, and another and probably better way of doing it is that some of the next generation sequencing systems that are coming up will be single molecule sequencing, which means they will sequence just a single molecule of DNA without amplification. And so they would be ideally suited to doing this kind of single cell whole genome sequencing. So I think it is quite certain, again, that in the near future it will be possible to get a complete sequence from one cell taken from an embryo. Okay, how expensive it will be, that's another issue. It has to be quite accurate if you want to use it for some kind of diagnosis, but things are going this way. I don't know how long it will take, but the idea is that then, to the extent that we can predict the phenotype of the child from the DNA sequence, and that extent is still limited, but it's going to, again, it's going to get better and better. Then it will be possible from the DNA sequence to have some idea of what the child will be like, what is his susceptibility to various diseases, what is his height at adult age, the color of the eyes, all sorts of things. For example, eye color can already be quite reliably predicted from the DNA sequence. It involves several genes, but we know which genes are, and we can predict which child would have blue eyes and which child would have dark eyes. And so you can imagine at least wealthy people going through this procedure and having sequence of 8 embryos determined and picking the child they want from this set and having that embryo implanted. It's not for today, it's not for tomorrow, but it's probably not so far in the future. And again, of course, then you have lots of issues that come up. So this is, I think, almost my last slide. So the message I wanted to give you is that the technology is moving very quickly, that there is demand, a commercially interesting demand from people, or in some cases, really justified demand, like non-invasive prenatal diagnosis. There are a number of difficult ethical issues, and this really needs to be taken into account in the discussions. So we try to avoid the situation, which I quite like. So you see the DNA double helix, and you see the science moving very fast, and ethics. I'm trying to keep up. Maybe to end on a positive note, yesterday evening we had the decisions from the Supreme Court of the US on the patents on the breast cancer genes. If you remember, there's a monopoly by a company called Myriad Genetics on doing the breast cancer test. This is what Angelina Jolie underwent recently and decided to have double mastectomy. And there was a big fight contesting the patents, and sort of probably final decision was reached yesterday, was announced yesterday by the Supreme Court. And it's a decision that, well, doesn't eliminate patents completely, but very much restricts where those patents can be taken. And actually, I think hours after that decision, there's already one company in the US that announced they would do the test for $995. And Myriad so far was charging almost $4,000 for the test. So it's not hopeless, but things are really moving fast. Thank you. Any questions? We have
10 minutes?
Okay.
Yeah.
10 minutes discussion. Is so we have mothers here.
Many
Ladies has already some children. Okay.
Auspicious fathers.
Father is evolutionary, nothing, you know. So what's your opinion about all those kind of testings? Okay.
One thing I never think about having a perfect baby. So when I was pregnant, I never think about having this. But however, yes, it's really a problem in the near future because I know that well, some of the anxious parents really want to have a quality baby. So yeah, so in the near future, we will see more and more stronger babies. So maybe Superman in the beginning. I don't know. Thank you.
So is it possible to have some information about the intelligence of the baby from those genetic informations or characters?
Yeah, it's
I think intelligence might be the first, especially can we determine the magnetic genes?
yeah. No, I mean that's the issue of complex diseases or complex traits, which means things that are clearly influenced by genetics, but where the influence is exerted through a large number of genes. So intelligence is strongly heritable. I think it's generally agreement that, okay, if you look at IQ, which is not exactly intelligence, it's one particular aspect of intelligence, but the heritability of IQ is generally estimated to be 50%, so which means that 50% of the variation within a group in intelligence is inherited, and 50% is due to circumstances. So in principle, it should. So if it's 50%, if this heritability, 50% heritability is somehow encoded in the DNA. However, finding the genes involved in intelligence, or genes involved in autism, or genes involved in even things like Crohn's disease has been very difficult, and people have found, with the very sophisticated so-called GWAS, genome-wide association studies, they have found genes that have an influence. So one version of the gene is associated with slightly higher risk of this disease or a tendency to intelligence, but the effect of each individual gene is very small. So if you wanted from DNA analysis to have an estimate of the potential intelligence of the child, we would have to know all the genes that are involved, and it's probably 1,000 or several thousand, and to know the influence of each variant, and to then use this information. We are very far from that. At this time, from DNA, you can have some idea about the risk for a number of complex diseases. Of course, you can find out the risk for Mendelian diseases. You can have some idea of eye color, skin color, hair color, risk of obesity, difficulty for cessation of smoking and things like that. But it's a lot less predictive than most people think. The general public tends to think that DNA gives you your fate in black and white, and that from DNA you can say you will die of a heart attack at 43 years and 8 months, and that's of course rubbish. What the DNA can tell you maybe is that your risk of heart attack is 5% higher than the general population. Yes.
Are there any more questions?
Not really a question, but I'd like to first thank Professor Jordan for his very informative talk, and secondly, I'd like to echo his worry for this widespread availability of genetic tests. And my reasons for sharing his concern is because I see there's a difference between how these genetic tests are offered. They're often offered by laboratories rather than through medical institutions. So they're compared to physicians that are doing diagnosis, they're less regulated. They're more like commercial enterprise, and that makes it more easy to be abused. And secondly, because they often offer it directly to consumers, I see the worries of Professor Jordan's even more, because now they're cheaper and there's more tests that you can do. So I do see that the urgency of bringing it under control. However, the third reason that makes it difficult to regulate is because it's often offered through internets across the borders. So I echo Professor Jordan's concern about these genetic testing. However, I'm less pessimistic about the area of PGD because PGD is offered through before the embryo is even implanted. That means you have to do an IVF, in vitro fertilization, before you can test the embryo. And IVF are, well, first of all, that means for a woman that could have naturally conceived, now she would have to undergo more risk through IVF. So that means she'll have to weigh whether it's worthwhile to undergo and give up the natural conception and undergo all the risk of retrieving the eggs and probably lowering the chance of fertilization and also all the costs of IVF, in addition to PGD, even though PGD is, but that means it's normally done in the clinic, which has, so I'm more hopeful of PGD, but I like to echo Professor Jordan's worry about genetic testing. Thank you.
Yeah, well, I understand your argument, Wendy, but first, I think that the tendency, okay, so to have fewer and fewer children, and the mother's age is going up, and so more, and of course, if the mother is around 40, the natural conception is not very successful, and so many of these mothers actually go through IVF anyway. And so that's one tendency which I think is a really strong tendency everywhere. And some people, I mean, argue that in the future it may be considered irresponsible to start a pregnancy by natural means, as it is considered more or less irresponsible now to deliver at home. which used to be the common practice like 50 years ago. Now, almost all deliveries occur in the clinic or in the hospital. And maybe 20 years from now, at least in rich societies, it will be considered irresponsible to have just any child that comes by random events, is when you have the possibility to select a good one through this procedure.
Yeah, I have two questions for Professor Jordan. This progress give us the image that these things can be done for fancier reasons. But the same technology probably in certain culture, certain society will be used for a very traditional, simple reason, simple causes. For example, I can expect that maybe for this kind of fast screening technique, there are probably more couples they are concerning to use this for only for the sex selection detection. So the old issues, okay, with the new method to do that will change the scenario, because currently, okay, even the government, they are, for example, the Bureau of Health Promotion, they are quite concerned of the sex ratio. Okay, they have been monitoring and try to intervene certain like the obstetrician clinics. But in the future, the couple can simply avoid this. Okay, they can go to simple lab or do the DTC method. So at the same time, when we are thinking about the more complex traits, but for these kind of traditional or even cultural urgent issues, we probably need to think it again.
Yeah, because I guess some sex determination by ultrasound can only be done later. So the determination is more traumatic.
And that leads to the next question probably we should all concern. So for all those new progress, and for example in Taiwan, so who should be watching or who should be discussing? It should be from the medical profession itself, or maybe we should expect that the pharmaceutical company also need to have some code of practice, which ethics should be emphasized a lot. And what kind of process or for discussing and having some practice guideline? What is the governmental roles? I think that's probably more important.
Yeah, I agree with you.
I think the very important thing I like to bring up is, actually, the maybe come up with different characteristics like height, weight, or intelligence, or disease, or whatever. Okay, that's the phenotype. Okay, but what determines the phenotype? Of course, the genotype is very important. So now, with all those techniques, we can know the genotype very, clearly and very comprehensively. But you should know that from genotype to phenotype, there's two very important factors. The one is environment. Okay, they will affect the process of development. Okay, even the same phenotype, when the environment is different, the phenotype will be different. Another thing is stochasticity. That is the chance. Okay. So I think you must be. I think the very important thing to know is to convey to the patient, to the parents, to the public is even if you know all the genotypes, you cannot very sure about phenotype, because there is a very complicated process of development. Okay, up to now we did not know where is the development information located in the genome. Nobody knows. That's the dark matter of the genome. Okay, and of course, if you have some infections, if you have some anything else like what we eat very much now, the 毒淀粉, okay, it might be in somewhere influence this because. For me, I think the most difficult, most challenging discipline now, as far as I know, is the development. Okay? And of course, there's a chance. There's a chance. Okay, there's always a chance. So one day, they asked me whether we can tell, because there's monozygotic twins. He said, how can you differentiate to monozygotic twins? I think I told her that you just look at the fingerprint. Your fingerprint is random, okay? It's not determined by your genotype. Even if they have the same genome, the fingerprint is not different. It is different. So I think you must have a very, because now we think that as long as we know genotype, we can predict the phenotype. Of course, in terms of a single mutation, single gene disease like thalassemia, cystic fibrosis, yes, it can be done. But for most of our phenotype is complex trait. So this is very important. So we must convey this. I think today is a very good occasion because you are very highly concerned about this issue. Genotype is not equal to phenotype. Okay, so I think this must be a priori information. Okay, is there any more questions? No, let's take a break. Okay.
We have a 10 minutes break. The next session will be from 20 past 10. 我们邀请其实坐在外圈的尽量坐到内圈来，就是这边有比较舒服的椅子，大家请不要客气。
My father and my uncle were all of them. But one has 7 children. Another has no children.
That's a conservative idea.
Yeah, sure, but then, 哎，好，
老师好，我现在在医研部，医学研究部，对，院病理，对，谢谢。